Okay, our next speaker is going to talk us through the project Metabolic and Enzyme Engineering for the Biotechnological Production of Partially Acetylated Chitosans, or ChitoBioEngineering for short.
Yes, this was. Okay, first of all, I'd like to thank the RIB team for organizing this final conference of our batch of projects. For us, for our project, it's a pre-final report I'm giving. I'll tell you in the end that we got an extension so that the project will be running until the end of this year. We're interested in chitosans, so molecules, polysaccharides and oligosaccharides produced from, which can be derived from chitin, one of the most abundant biopolymers on Earth. And chitosans are typically generated from chitin, which is extracted from shrimp or crab shells. And then this chitosan can be used in many different applications, as you can see here, in the material sciences and biotechnology. But what is most interesting to us are applications in life sciences. Chitosans have a broad variety of very promising biological activities, and they can be used, therefore, in many different potential applications. One of them, well-known potential application, is plant protection. Plants which are treated with chitosan may become more resistant to disease. May, it means it doesn't always work. Sometimes it works, sometimes it doesn't work. And unfortunately, the same is true for the biomedical activities of chitosans. You can generate chitosan gels, which can be put onto wounds, can be used for wound dressings. And under these chitosan-based wound dressings, sometimes, even on large scale and deep burns, a completely new skin can develop, which even has the original pigmentation again, without any formation of scars. It's an almost more miraculous process, but unfortunately it doesn't always work. And so our work for a long time has concentrated on trying to understand structure-function relationships for the bioactivities of chitosans, trying to understand which chitosans exactly are important for which biological activities. Chitosans, as you can see here, are linear molecules of only two different units. This is the acetylated glucosamine unit, which makes up chitin, and then the deacetylated glucosamine unit. So there are only two different units in this molecule. And when we started to think about structure-function relationships, we first decided to concentrate on the degree of polymerization and the degree of acetylation, and their influence on the physical-chemical properties on the one hand, and then on the biological functionalities on the other hand. Degree of polymerization would mean the number of units involved in the molecule. So in this two-dimensional chitosan matrix, you would have the degree of polymerization here, where we start with monomers, then oligomers, small polymers, and large polymers. And on the x-axis, you would have the degree of acetylation. Fully acetylated would be here, so chitin would be up in this range. Chitin is insoluble. And then by partial deacetylation, you can reduce the degree of acetylation to obtain different types of chitosans. So in the first project we had, a European project named Carapax, we were interested in analyzing the influence of degree of polymerization and degree of acetylation, and we generated a number of chitosans differing in these parameters, and these chitosans were generated using chemistry. So by chemical deacetylation, chemical depolymerization, chemical reacetylation, we can move around in this matrix, but the important aspect of all the chitosans produced in this way is that the pattern of acetylation will always be random because of the chemical processes involved. So in a second project, we decided, because we found that controlling dP and dA is not enough, we decided to also try and look at the third dimension, the pattern of acetylation. And we showed proof of principle that using enzymes we can generate chitosans with non-random patterns of acetylation, blockwise patterns or more regular patterns than random patterns. So this was in the second of our projects, and then the next project focused on identifying novel enzymes, novel chitin and chitosan modifying enzymes to increase the diversity of chitosans with non-random patterns of acetylation. These enzymes were, of course, characterized in detail, and then the novel novel enzymes were used to try and generate novel chitosans, which are now in the process of being evaluated for their potential. As the result of these 3 European projects, we started the first public-private partnership in the German project, which is called the Cookie BCA project, where the Chi stands for chitosans, our very specific defined chitosans. And we could show that a combination of 2 different very specific chitosans, one used for seed coating and one used as a foliar spray, can have tremendous effects on crop plants, here on tomatoes, where we can induce disease resistance, we can induce stress tolerance. That was an unexpected side result. When we grew these, when we tested our plants in Morocco, in the greenhouses of the king, there was a problem last summer that in one of the greenhouses, the air conditioning stopped working because there was a power failure. And so the temperature in the greenhouse climbed to over 60 degrees. And after 2 hours, when the power was reinstalled and air conditioning came, almost all the plants in the greenhouse had died, except for a single row of plants, which was treated by our chitosans. So apparently the chitosan-treated plants have a lot more and stronger developed roots, which allow them to tap water which is not reachable by normal plants, by untreated plants. So we have a yield increase even in the absence of stress, and we have stress tolerance and disease resistance. You can see here in this experiment, which we had to terminate because the plants grew too large and there was no space in our small greenhouse in Münster. We had about 200% increase in yield. So this was extremely successful. And as you can see, these 3 European projects led to this applied project, which is very successful now in agriculture. And all the chitosans we used here were polydispersed chitosans, which means these chitosans which came from animal origin and which are always mixtures of different chitosans. Now, in the current project, we are trying to improve on this by moving towards the biotechnological production of chitosans, and now not mixtures of chitosans, but monoclonal chitosans, where we would have a single bacterial clone, which can produce a very specific chitosan oligomer of defined architecture, which we can then try and use in different types of high-price applications. So the aim of our ChitoBioEngineering project was to set up biotechnological production for these defined chitosan oligomers, which should be defined in terms of the degree of polymerization, the degree of acetylation, the pattern of acetylation. And these should then be used for different applications. And of course, the hope is that using these well-defined chitosans, we would have well-defined, reliable, reproducible bioactivities. And the approach used in this project is to express chitin synthase genes and chitin deacetylase genes in suitable host strains so that they can produce chitin oligomers and then deacetylate them to chitosan oligomers. And then, of course, we have to engineer these strains for optimal performance. So we had 3 academic partners, ourselves from the University of Münster, involved in genetic engineering, identifying suitable genes and doing the first expression of these genes to see whether they can be used for the in vivo production of chitosan oligomers. Then Barcelona for the protein engineering, trying to optimize the enzymes we have to produce different types of oligomers or more precise oligomers if they're not precise enough. And then the third partner, University of Ghent in Belgium, is responsible for the metabolic engineering of the host strains for maximum yield and maximum optimum purity of the products. Then we have two industrial partners, the Biobase Europe pilot plant again in Ghent, which is responsible for upscaling to the pilot scale so that we can get larger amount of some of our oligomers, which can then be tested by our second industrial partner, Heparin Medical Chitosan from Germany, who use these chitosans in trial applications with their partners from industry, mostly from cosmetics and veterinary medicine. State of the art at the beginning of our project was that a certain bacterial chitin synthase gene, the NOT-C gene, had been expressed in E. coli and produced the chitin pentamer. And then if the NOT-B gene, a very specific chitin deacetylase, was added, one type of monodeacetylated chitosan pentamer could be produced. So this was the state of the art at the beginning of our project, one chitosan oligomer which can be produced. Our approach now was to use different types of chitin synthases and different types of chitin deacetylases to try and increase the diversity. And then, of course, to optimize the strains, the enzymes, and the pathways to have optimal production facilities. The targets we were trying to reach were firstly different degrees of polymerization. So not only pentamers, but also smaller or larger ones. Different degrees of deacetylation, not only monodeacetylated ones, but also multiply deacetylated ones. And finally, different patterns of acetylation. So these would be oligomers with the same degree of polymerization, the same degree of acetylation, but having the deacetylated unit, for instance, in different positions. So these are our targets. And at the midterm review report in Düsseldorf, I could present that we were able to produce in vivo tetramer and pentamer and a little bit of hexamer also. And we had not only the monodeacetylated ones which had been described previously using the NOT-B gene, but using a different chitin deacetylase, we were also able to produce a different monodeacetylated one, but not the unit at the reducing end, but at the non-reducing end, but the one next to the non-reducing end is deacetylated. In vitro, using our enzymes in vitro, at the time we were able to generate a few more different types, different oligomers, a third position, so a third pattern of acetylation for the monodeacetylated one, but also several enzymes which are either several oligomers which were fully deacetylated or partially deacetylated, but having different degrees of acetylation, but always a specific pattern of acetylation. This was our in a way our surprise, of course, is what we had hoped for, that chitin deacetylases would generate different patterns, and so far it is true that almost every chitin deacetylase we try successfully will give us a different pattern of acetylation compared to the previous ones. So this was at the time of midterm. Now at this time when we are almost finished with the project, we can generate dimers. All 4 different dimers are possible to produce. Out of the 8 possible chimers, we can produce 5 different ones, as you can see here. If you go for the tetramers, out of the 16 possible tetramers, we can produce 12 different ones. There are still a few missing. We have, of course, tried to achieve those also. For instance, you can see here, if you look at this, there's the 4th one missing, where we would have only the reducing and deacetylated. This is a tough one. We had a very good idea, a very good lead, but unfortunately, so far, it's not active. So this is missing, and this is also why some of the others probably are still missing. So a few challenges remain, but the diversity has dramatically increased compared to what was available at the beginning of the project. Also, we have a selection of different pentamers, and in principle, we can also generate the hexamers. The problem there is that in vivo, hexamer production is still very low, most likely because of the insolubility of the fully acetylated chitin oligomers. At DP6, they are still soluble to some extent, but if they grew larger, they could become insoluble. And we guess that the DP6 already is too insoluble to be produced in vivo in good quantities. Now, if we look at the upscaling for the pentamer, for the fully acetylated chitin pentamer, we have upscaled it in a 100-liter fermenter, and we came up with about 1 kg of the material, of 1 kg of pure, very pure chitin pentamer. If we look at it in mass spectrometry, which is extremely sensitive, we see only the pentamer after downstream purification. This is better than anything you can buy on the market, which is at maximum 95% pure. And if you see the price of what you pay in the catalog, and you start calculating this, unfortunately, there is no market for 1 kg of the pentamer. Now, this pentamer, of course, can be used for deacetylation. This is what we've done so far in better quantities only in vitro. So we have generated these 3 different pentamers in vitro in about 50 milligram batches, which are also extremely pure and which are now being investigated in different applications. The problem here is we can produce them in vivo also, but the downstream processing of the partially acetylated ones is much more challenging than of the fully acetylated ones. And so far we do not have a process which yields good amounts of high purity chitosan oligomers. Now, what's it all for? This is a picture I've been showing for a long time. The idea behind the chitosan oligomers in plant protection would be that plants are used to see chitin. Chitin is part of fungal cell walls. Chitin is part of insects' exoskeletons. So when a plant sees chitin, it knows that something is wrong, and then defense reactions are started. Typically, the plant will stop all other activities. It will stop photosynthesis. It will stop producing starch, which we wanted to do, and it will only defend itself. So this may be successful in pathogen defense, but it's not a good strategy to treat plants with this signal molecule, because if they would stop photosynthesis, they would stop producing what we want them to do. Instead, if we give them a specific chitosan oligomer, then it looks almost like chitin, it looks almost like a pathogen, but not quite. So the plant will be on alert. It will still keep going, it will still do photosynthesis, will still produce starch, but it will prepare it for attack. And if a fungus or a bacterium then comes, the plant is ready for defense and can defend itself more efficiently. Now, I always said that this is the theory. We have evidence that this is true, but we have no idea how this molecule looks like. We know how to produce it, but we can only produce a wide mixture of many, many different chitosan oligomers where we know the one or the few which are having this activity are present, but we don't know which ones they are. And typically, at this point, I would say we don't know them yet, but we hope to know them in future. But even if I would know them now, I wouldn't tell you. The second part is still true. We have found some which are active, and I'm showing you one example here. This is actually the only experimental slide I'm showing, in which I show the result of an experiment, but I have to tell you it's faked. This is not the true result, meaning the oligomers I'm indicating here are not the true oligomers. I'm of course not allowed to tell you which ones they are. This experiment is a priming experiment in cell culture. Rice cell cultures are treated at this point, with a chitin hexamer which is acting as an elicitor. It triggers resistance reactions. The resistance reaction can be monitored by the production of hydrogen peroxide in the medium. So when we add a large amount of this chitin hexamer, you can see this transient increase in hydrogen peroxide and then the decrease again, an oxidative burst. This is the defense reaction triggered by chitin. If we use low concentrations of chitin, then there's a very small but almost imperceptible oxidative burst. Now, if we pre-treat the cells 3 hours prior to the elicitor treatment with something which has priming activity, then you can see this is the one. This is a partially acetylated chitosan oligomer which has priming activity. Now, even if we add this at very low concentration, if at time point 0 we add elicitor at also a very low concentration, at the concentration which alone would not induce much, we get a very strong induction of the oxidative burst. So the pre-treatment of these cells with low amounts of the chitosan oligomer put the cells into a state of alert so that they are able to see very low amounts of the elicitor of the molecule indicating the presence of a pathogen, so that a very strong resistance reaction can be mounted. If we use a different chitosan oligomer, again, this is not the true one, but one which it has the same degree of polymerization as the active one, the same degree of acetylation as the active one, but a different pattern, it's completely inactive. Even if we use it at high concentration here at the time of priming, there's no effect on elicitor activity. So yes, the pattern of acetylation matters. This is the first proof that the pattern of acetylation in a chitosan oligomer is responsible for the biological activity. So this, of course, is extremely important, because it means, as a proof of principle, that it's worthwhile to go all this long way to try and produce specific chitosan oligomers with a specific pattern of acetylation, because the pattern does matter. This was only a hypothesis at the beginning of the project, and it's an experimental truth at this time.